We're going to be spending time talking about the liver, an extraordinarily important organ, the largest gland in the body. It has both exocrine and endocrine functions. Exocrine in the sense that it makes bile and albumin, and endocrine in the sense that it makes a host of hormones, which I trust Dr. Whittingham has spoken to you about in connection with the digestive function. Before we talk about the histology, there is a gross anatomical feature of the liver that is extraordinarily important in the construction at the histological level of the liver that I'd like to remind you about. And that is that the liver receives blood from two different sources. In other words, it has a dual blood supply. First is that there is oxygenated blood that is brought to the liver directly via the hepatic artery. Secondly, blood also arrives in the liver via the hepatic portal artery. And this is an artery that is carrying newly absorbed foodstuffs from the digestive tract. Everything that you've eaten ultimately ends up being transported to the liver directly, except for lipids. Proteins, carbohydrates, and the like are broken down partially in the gut, and then that is carried to the liver. So, did I say portal artery? I meant portal vein. Two different sources, hepatic artery and hepatic portal vein. Both carry blood to the liver. And you'll see how this turns out when we look at the histology of the liver. Yes?
So the hepatic portal vein is deoxygenated?
Say that again?
The hepatic portal vein is deoxygenated?
Well, it's venous blood.
Okay.
So there probably is some oxygen there, but it's relatively low. Okay, in terms of general functions of the liver, and we just said that it receives all newly absorbed foodstuffs except for lipid. So it's an extraordinarily important organ in the digestive process. Secondly, the liver is the site of detoxification of many, many different substances. For example, deamination of amino acids occurs in the liver to ultimately produce urea. Liver is also the site of production of blood proteins, mainly albumin. And a very important function is that the liver acts as a storage site for glycogen, which is a form of carbohydrate. And at almost any given time, you're carrying around several pounds of glycogen in your liver. Now, in order to study the histology of the liver, one, of course, has to make sections. Now, if we look at human liver, the landmarks are very difficult to make out. Instead, what is usually done is that we look at the pig liver. Pigs, bears, raccoons, all have livers in which the basic structural components are very clear. So if you understand what is going on, for example, in the pig liver, it's relatively easy to extrapolate it to the human condition. Okay, now the basic structural unit of the liver is called a lobule. And lobules, if they're cut exactly right, clearly have a hexagonal shape. So let's just a lobule. In the center of each lobule, there is a blood vessel. And this blood vessel is called a central vein. Now, don't let that name confuse you. It's really not a vein in the classic sense of the term. If you had read in your book about the structure of veins, you would know how they're put together. They consist of multiple layers of different kinds of cells. The central vein really is, it just consists of simple squamous epithelial cells. It does not have any of the muscle or the usual connective tissue that we would associate with the vein. It is called a central vein, but again, histologically, it does not resemble a vein. If you look further, you will see that at each one of these corners, there is a complex collection of structures which we'll come back to and talk about in a few moments. These are called portal areas. So each central vein then is surrounded by 6 portal areas. What makes these lobules relatively easy to see in the pig is that there is connective tissue around each lobule. So it makes each lobule stand out. The major difference between a pig liver and a human liver is that we don't have that connective tissue around circumscribing each lobule. So when you look at a human liver, you almost have to imagine that that structure is present. However, from a functional standpoint, it doesn't matter. There are lobules in the human liver as well. Okay, now let's look more carefully at the structure of a lobule. When you look at low magnification, what you seem to see is that there are radiating columns of cuboidal cells like so. And these structures are called liver cords. Turns out that the interpretation of liver cords is completely wrong. That you do not have cords of cells that make up the lobules. In between these so-called pores are blood spaces, which have been called sinusoids. Okay, so it was originally thought, way back in the 1800s, that the liver lobule consisted of cords of cells, and in between were these vascular spaces called sinusoids. It wasn't until the 1940s that careful reconstructions in 3 dimensions were made of the lobules. And it turned out that that original interpretation was wrong. Instead, the lobule is sort of like a Swiss cheese. You imagine a big chunk, hexagonal piece of Swiss cheese that has these holes in it, and these holes are irregular and sort of tunnels in the chunk of cheese, but that they all sort of make their way to the center of that chunk. Okay, so in 3 dimensions, actually, there are no cords. There are sinusoids that are irregular in 3 dimensions, but inevitably make their way towards the central vein. Now it turns out that every cell, every hepatic cell, liver cell, in the lobule borders on at least one sinusoid. So every liver cell is exposed to a sinusoid. Everybody with me so far? Okay, that's not too difficult, but it took almost 100 years for this concept of the structure to be established. We now know it's absolutely correct. Now, if we look more carefully at one of these so-called cords, remember they're in 3 dimensions, they're running in a lot of different directions. What you would see, I hope that the chalk does better on this. Of course it doesn't. If this is a central vein, and here is a sinusoid, each sinusoid opens into the central vein. If we follow the sinusoids back far enough, we will see that ultimately they have 2 different origins. That in the portal areas, there are branches of the hepatic artery, and there are branches of the hepatic portal vein. And you see both of these blood vessels empty their contents into the sinusoid, and the blood mingles together and is carried ultimately into the central vein. Okay, you got that now? In the portal areas then, there are branches of the hepatic artery, branches of the hepatic portal vein. These empty their contents into maybe the same sinusoid, and that blood is carried through the sinusoids, ultimately to empty into the central vein. And since each hepatic cell has at least one surface that faces a sinusoid, each hepatic cell receives oxygenated blood and blood carrying newly absorbed foodstuffs. Okay, this is sort of a complex idea. But now, let's take a look at a sinusoid more carefully. Each sinusoid, of course, is surrounded by hepatic cells. And so on. If we look carefully at the interior of the sinusoid, it becomes obvious that there's a blood vessel inside it. The blood vessel has the general appearance of a capillary. Yes?
It's not clear the difference between a sinusoid and a lobule. The lobules are inside the sinusoids, or the sinusoids
Just the other way
are inside
around. You got it turned around. Lobule is the big structure that is permeated by sinusoids. And now we're looking at a single sinusoid. Within that sinusoid, there is a capillary. That capillary consists of discontinuous epithelium. Remember what that means. It means that the cells don't fit together absolutely perfectly, that there are spaces between adjacent cells. These are not big spaces. It would be akin to if you had a jigsaw puzzle and you had two pieces that really didn't fit together, they didn't belong together, but you jammed them together. There would always be a little space between the processes on one piece and the recess on the next piece. So there are these little slits in the epithelium. Now, you notice I drew this with a space between the endothelium of the sinusoidal capillary and the hepatic cells. There really is a space there, not an artifact, and that space was discovered many years ago, and it's called the space of Disse. Disse was a German histologist who first described that space. I have a friend who claims that this was discovered by an Italian graduate student who saw it and said, "What's Disse's space?" Disse was a German, and it's a real space. If we look at the space of Disse, it turns out that it's not empty. There's all kinds of structures in there. First of all, the surface of the liver cells that faces the space of DISTA is covered by microvilli. So, see if I can sort of indicate that here, like so. So there are numerous microvilli of the hepatocytes there. Think about this. There are slits in the endothelium which permit blood proteins and blood, every part of blood except the cells of blood, to enter the space of DISTA. They don't have to go through cells. That material can go between the cells into the space of DISTA. But those slits are small enough to exclude cells. So red cells cannot get into the space of DISTA. Just the fluid contents of the blood. That means that the hepatocytes that border on the space of DISTA are exposed to oxygenated blood and to blood that contains the newly absorbed foodstuffs. Conversely, these cells can release their waste products into the space of DISTA, and that material will make its way into the capillaries that are in the space of DISTA. Turns out it's a lot more complex than this. It was originally thought by a man who was really critically important in determining the structure of liver, a man named Kupfer, that there were cells, part of the endothelium, that had phagocytic properties, that these cells were able to pick up material coming from the, getting into the blood and phagocytosing this material and destroying it. It was thought that Kupfer cells, as they were originally called, were part of the endothelium. Later studies showed that there were cells, phagocytic cells, that actually spanned or crossed the capillaries. And these cells are cells which now bear the name of Kupfer cells. They're not endothelial cells. They are basically macrophages that have taken up residence in the capillaries of the sinusoids. If one wants to demonstrate these cells in an experimental animal, very simple. All you have to do is to inject some kind of particulate material into the bloodstream. A very nice tracer is India ink. India ink consists largely of tiny carbon particles in a fluid medium. If you inject that into the blood vessels of a rat, the Kupfer cells will pick up the carbon particles and sort of self-label. So there are phagocytes that are present here. Now, it turned out that there was another cell type associated with the space of Dissa. These are cells that reside in the space of Dissa. They had been originally noted by Kupfer, and he sort of discounted them. He thought they were part of this phagocytic array. Actually, they were rediscovered about 1950 by a Japanese histologist named Ito, and he described these cells in a series of very important papers that were sort of first received with a great deal of attention in the West and then completely forgotten. And nobody remembered these cells, which were called Ito cells, for many years. And it took about 50 years, and another Japanese worker rediscovered them. And his name, not important, named Wake. Wake wrote some papers in the Western literature, and suddenly these cells became very important. And because of their shape, these cells are called stellate cells. So stellate cells are a different kind of cell that reside in the space of Dissa. What do stellate cells do? Well, it turns out that they can store certain kinds of lipid. The lipid that they're most attracted to is vitamin A. If you want to demonstrate Ito cells or stellate cells in the liver of an experimental animal, you need only inject excess vitamin A into the bloodstream, and suddenly these cells are filled with lipid droplets. They show up very, very strongly. So a major function of these cells is storage of vitamin A. They're extremely prominent in some animals. For example, the fish, the cod. I guess it's not fashionable anymore, but when I was a child, most children were given cod liver oil. I'm sure you've heard of that. Cod liver oil is nothing more than you just take liver of cod and squeeze it, and it expels the vitamin A that has been stored in these numerous Ito cells that are present in the cod liver. So it's a source of vitamin A. Knowing where to look, it became possible to design specific stains for these cells. And it turns out that if you use a stain that contains gold salts, these cells are stained sort of a blackish color, at least the droplets in the cells are. So we say that these cells are aureophilic. That's A-U-R-E-O, philic. Aureo, referring to gold from Latin, French. Yes?
So are the cells staining black, or is the vitamin A staining black?
It's a good question. It's probably the stored vitamin A, because even if you don't experimentally put a lot of vitamin A into an animal, we all have vitamin A in our body. So these cells will be labeled. Now these cells do something else, it turns out. These cells also are involved in the production of reticular fibers. Reticular fibers are found all through the space of DISA. I hope you remember what you learned about reticular fibers. They're really a form of collagen, and that they are very delicate compared to standard collagen fibers, and they form what amounts to the connective tissue framework of liver. Liver does not have huge amounts of collagen in it. Its framework consists of a sort of delicate network of reticular fibers. Just think now, when you eat a steak, the meat sort of fights back because there's collagen in it. In contrast, liver has a kind of a mushy feel because it has relatively little collagen in it. What collagen is present is in the form of reticular fibers. And it turns out that the stellate cells make those fibers. So these cells are very important functionally. One, lipid storage. Two, reticular fibers. These cells are not independent agencies. They have gap junctional connections to the endothelial cells, to the hepatic cells, to even Kupffer cells. They all work together in a kind of a grand symphony of cellular function. Sir?
Where's the difference between the stellate cells and fibroblasts then?
Fibroblasts are completely different cells. Fibroblasts are connective tissue cells that make collagen. These cells make a form of collagen, a very delicate form, reticular fibers. There's a big difference between the two. There's no loose connective tissue here, none. These are the collagen-producing cells.
It's not exactly the same.
They're not fibroblasts. They have a completely different origin. Okay, yeah. Before we leave the sinusoid, I have to mention there's another cell type that is occasionally found in the space of DISA. These are cells which I don't want to get into in any great detail, but you've probably heard about them from Dr. Weinberg. These are a kind of lymphocyte that are known as natural killer cells. These cells are occasionally present in the space of DISA. And I don't want to talk about those cells because it's really immunologically important stuff, but it doesn't really concern us. So you can see the space of DISA is a busy structure. It's got microvilli, it's got stellate cells, it's got natural killer cells, it's got reticular fibers, and it's all arranged in such a way that the contents of the bloodstream, with the exception of the blood cells, can get into the space of DISA so that each hepatocyte is exposed to oxygen-rich and foodstuff-laden blood and waste products can be flushed out by the same route. Okay, so it's a complex system, and it took a long time to figure out exactly how this works. Now, it turns out that in addition to blood flowing from the periphery of the lobule towards the central vein, that there is a countercurrent stream of fluid going in the opposite direction. And how can I indicate that? In between the adjacent hepatic cells, there's another system of channels. And the system of channels exists wherever two hepatocytes are together next to one another. So two adjacent hepatocytes will tend to form these channels. These channels are called bile canaliculi. This bile, which is a secretory product of the hepatocytes, flows into these canaliculi, which ultimately open into the third structure that is present in a one of these parietal areas around the outside of the lobule. Remember, there's a branch of the hepatic portal vein, a branch of the hepatic artery, and then there's a branch of the bile canals that's present. There are always those three structures called a portal triad because they're visible. It's a bad name because actually there's also a lymphatic capillary there, but it's very hard to see and was overlooked for many, many years. So it was called a portal triad. It really should be called a portal tetrad, but nobody calls it that. Very important now, the bile flows in a countercurrent direction. So blood flows centrifugally towards the center, and bile flows centripetally towards the periphery of the lobule. Very important is that this bile is separated from the bloodstream. In other words, there's no way for bile to get into the blood because the cells are very close together and the canaliculi are sealed by tight junctions. If bile somehow escapes the confines of the canaliculi and ends up in the blood, that's a very serious condition, and it produces what is known as jaundice. It produces a kind of yellowish cast to the skin because the bile has a sort of a brownish, greenish, yellowish color. And it depends on the integrity of those tight junctions to keep bile from mingling with the blood. If bile does, in fact, get into the blood, as I said, it leads to this serious condition called jaundice, and very difficult to treat, except one hopes that the liver can regenerate itself and produce new tight junctions.
Tim?
Yes,
Jaundice was related to bilirubin, not bile, or is the same thing?
it is, what, say that again?
I thought jaundice was related to bilirubin, not bile, or is it the same thing?
Bilirubin is a content of
Okay.
bile. There are a number of different pigments that end up in bile that could end up in the bloodstream. Okay, now, I just mentioned the word regeneration. Now, liver has a remarkable capacity to regenerate. One can literally take 75% of a human liver out, transplant it, and although it takes about a year for the liver to completely regenerate itself, it will. It can do it. In other words, if you leave just 25% of the original liver within a year, and it's sort of a kind of a rapid curve at first, and then it slows down. But the liver is capable of regeneration, which is why you read about people receiving liver transplants. Without a functional liver, you cannot thrive. I mean, one's life would be very limited. Yes?
Does it matter what piece of the liver you transplant, or can you take any piece and it'll grow back?
That's a good question. I'm not up on that thing. I would imagine that whatever part of the liver is left behind is capable of regeneration. Yes?
Also, does regeneration differ by age or by gender? Like, as you grow older?
I'm not aware that that is a major factor in regeneration. I mean, that goes way beyond the scope of this course, but if you're interested, I'm sure there's a vast literature on liver transplants and liver regeneration. Okay, now, the last thing that we need to talk about are the liver cells per se, that is the hepatocytes. And these are really quite remarkable cells. When you see them in a light microscope slide, what you see are cuboidal epithelial cells. And they have one nucleus, but a certain percentage of liver cells have two nuclei. They're binucleate. The significance of such cells is really not understood. And here's a little mind experiment that you can do. If you've got a cell with two nuclei, how does it divide without getting everything all mixed up? Think about that. I'm not going to answer that question for you, but something for you to puzzle about. If you examine these cells, first of all, turns out they are loaded with mitochondria, as you would expect in a cell that has so many important functions. It is estimated that in a single hepatic cell, there are about 1,000 mitochondria. This is why the liver is the favorite organ for biochemists who work on mitochondria. One can take the liver of one rat and get out a pellet of pure mitochondria. A little bit more difficult with mice. You usually use 2 or 3 mouse livers to get out mitochondria. But huge number of these mitochondria. And these mitochondria are turning over at a relatively rapid rate. They have a half-life of something like 10 days. So you start out with 1,000 mitochondria. 10 days later, only 500 of the original mitochondria are left, and you got 500 new ones. So it's like churning these mitochondria over and over again. Now, if you look further into the cytoplasm of these cells, well, remember I told you that they make blood proteins and albumin. Well, to make proteins, you have to have rough endoplasmic reticulum. And of the cytoplasmic volume, now you don't have to write down any of these numbers or percentages, but I just want you to get an idea of just how busy these cells are. These cells have a lot of rough endoplasmic reticulum to make the blood proteins, and because they are involved in detoxification, they have lots of smooth endoplasmic reticulum. You may recall I told you that one of the ways that drug companies now look for toxicity of new reagents that they may have developed is simply to inject mice with this new drug and look to see if there has been an evolution of smooth endoplasmic reticulum. And if there are problems, there's usually a huge development of SER. Now, in addition to detoxification, as I told you, the SER is also involved in glycogen metabolism, and associated with the membranes of the smooth endoplasmic reticulum is glucose 6-phosphatase, an enzyme that you should remember, which is involved in the synthesis of glycogen and in the breakdown of glycogen. If you wanted to examine this, if you take, let's say, a mouse, feed it, look at it, after a few hours, look at the liver, you see that the liver is loaded with glycogen particles. Now you starve the animal for a day or two and look at it, all that glycogen is gone, but the SER, of course, remains. In addition, there are a number of organelles that we really haven't talked about in great detail. Of course, one of them is lysosomes, and those, I hope you remember, are involved in the breakdown of either phagocytosed material or the destruction of parts of the cell that have become senile and outlived their usefulness. And in fact, we knew about these structures, which we now call lysosomes, for many, many years, but nobody knew what they were. Another organelle that was discovered in liver is an organelle called a peroxisome. Peroxisome. As P-E-R-O-X. isome. Peroxisomes are involved in lipid metabolism and in detoxification. Their discovery was very important, and in fact, in some animals, they contain the enzyme catalase, which, you know, breaks down peroxidases. If you were to buy some catalase from a supplier, a chemical supplier, and just took the powder and looked at it in the electron microscope, it would consist of little crystals which come from peroxisomes, pure catalase. Okay, so obviously a great deal of the volume of a liver cell is occupied by these different organelles. And again, don't write these numbers down, but there are about 1,000 mitochondria. This is in one cell. There are about 500 peroxisomes in one cell. Anywhere between 100 and 250 lysosomes. There are millions of ribosomes that are present. One pair of centrioles, important for cell division. Your liver cells will divide in the course of your lifetime probably at least 40 times. So cell division is an important process in the liver. And then there's a huge number of microtubules and microfilaments, but of undetermined number. So these are really busy, beautiful cells. Tell you what, let me show you some pictures and we'll be finished rather than taking a break and have you come back for just a few minutes. So can somebody operate that projector for me? Which is that top button that flips forward. Anybody know how to operate an old-fashioned projector?
He's up here doing it.
It's on the side. It's not a button.
Yeah,
Okay.
yeah.
Okay.
It was weird,
Well,
but
it's
it
out of
wasn't
focus.
in Blazing.
No, you got way past where I want to be.
I'll take it back, don't worry, one
You
second.
gotta go way back. Go back, go back.
There we go.
Okay, and now can you focus
I'll focus it.
it's that thing right up front there.
It's good?
Oh, okay. This is from a pig liver, and what it does is it shows you very clearly the structure of a lobule. You see, each lobule has a central vein, and these are the hepatocytes that seem to form columns, but remember, they're very irregular, and they make their way to the central vein. And you can see, can you focus this? It's still
Sure, I'll
a
try.
tad out of focus. Just a little carefully. Slowly. Oh, okay. You can
As good as
see
it gets.
here, for example, that sinusoids in some places go directly into the central vein. And the central vein does not have the characteristic structures of a real vein. And in the corner are these portal areas that don't show up so well here. I'll show you better pictures in just a moment. The next slide.
Okay. So you're on
And out of focus. Okay, you don't have to keep fooling with it. Once it's in focus, it's in focus. Here are what used to be called liver cords. You see, here's one with two nuclei. And you see, every single cell is exposed to a sinusoid. These streaks are the nuclei of the capillaries that are found within the sinusoids. The next slide, please. This is sort of a cross-section through a lobule, and you see these cross-sections of sinusoids, and you see that every single hepatic cell borders on a sinusoid. And again, here's a binucleate cell. Most of these nuclei are Kupffer cells, but I'll show you those a little better in the next slide. Oh, okay. Now, this is a slide in which the blood vessels were injected with some gelatin that contains a blue dye. And then, could you focus this? Is out of focus? Okay. What you're seeing are the sinusoids that have been injected with this blue gelatin, just to show you how they make their way towards the center where there is a central vein. And the next slide. This is one in which the bile canaliculi have been stained. And you can see that these canaliculi all extend between adjacent hepatic cells and make their way out to the portal triad. Remember, there are two systems of fluid movement within a lobule. One where blood is moving towards the center, and the second where bile is moving to the periphery. The next slide. This is a portal triad, and here you can see there is a branch of the hepatic artery over there. It looks like an arteriole. This is a branch of the hepatic portal vein, and this epithelial structure here is a bile canal. So those are the three major structures that are present in a portal area. The next slide. In this preparation, the liver was cracked open and looked at with a scanning electron microscope, and it shows surfaces. Here is a cross-section of a sinusoid, and then here is a bile canaliculus between two adjacent cells. And here, looking into a sinusoid, you can see the endothelium of a capillary, and you notice the discontinuities in the epithelium. The next slide is a higher magnification to show, on the one hand, a sinusoid with some RBCs, red blood cells, here, and then way over here, and this is a little out of focus. Can you focus here for a moment?
Yeah.
Yeah, right there is a bile canaliculus, and you see that they're quite separate, never joined. The next slide. In order to demonstrate Kupffer cells, as I said, you can inject the animal with some kind of particulate material. In this case, there was a stain, and here, these cells have phagocytosed the particles and show up very clearly inside the sinusoid. The next slide is a really remarkable micrograph. Next. This is from a cracked open liver. This whole structure here is a sinusoid, and spanning the lumen is this structure here, which is a Kupffer cell. And you can see how it is strategically arranged to catch any particulate material that may be in the sinusoid. This is truly a remarkable micrograph. The next slide. And here again, sinusoid bile canaliculi, always separate from one another, always. And here you can sort of make out the space of Dissa with the microvilli of the hepatic cells. The next slide is an electron micrograph of of normal liver, and here is a sinusoid in cross-section, space of Dissa filled with microvilli, nucleus of a hepatic cell, black material is glycogen, and numerous mitochondria that fill the cytoplasm. Again, this is the space of Dissa with microvilli in it. Don't have a picture of an Ito cell, unfortunately. I think that's the last slide. Can you turn the lights
Yes,
on?
that is.
Can you turn the lights on? Okay, well, I hope at this point you believe me when I tell you that the liver is truly an almost miraculous organ. It has multiple functions. It can restore itself if it's injured, and is a beautiful example of histology, structure, and function being expressions of one another. Are there any questions about the liver? Okay, I didn't say anything about the gallbladder. You can read about it. It's basically a very simple organ, and you know, it just stores bile, and you get along perfectly well without it. There are a lot of people walking around whose gallbladders have been removed for one reason or another. Okay, if there are no questions. let me